Yes. So first of all, for IDPs, the experimental data which is being used is slightly different than for structural data, which is traditionally what the 3D bioinfo does. In fact, we've already heard that this is at the interface between the molecular and cellular level. And this means that there are certain techniques which can be reinvented or used in a different way in order to provide knowledge for IDP specifically. So at the structural level, we're talking about things like circular dichroism, where you have only indirect evidence of what is going on for the protein. You can use FRET, so first resonance energy transfer gives you information about distances which are maybe moving. NMR can be used in terms of the chemical shifts, for instance, which do not produce the full 3D structure, and SACs likewise just for the shape. So for the interactions, then this is a big issue for IDPs, because there's a lot of interactions mediated by intrinsically disordered regions. There are different technologies. And also for the function, there are larger assays, things like even phase separation assays, which are more at the cellular level. So this means that there is a whole set of information which is not currently very well captured in the databases. Next, please. And so one of the goals for the IDP user community is specifically to develop a centralized knowledge base for IDP data. This is due to the fact that there is a lack of standards in the field, so this has to be homogenized, and there are many individual resources. You see a picture there of some resources throughout Europe, and these should all be pulled in together and brought to bear on a single resource. Moreover, and next slide, please. Here you have a motivation for why a central database is also necessary. So these are two graphs showing you the overlap. The Venn diagrams show you the overlap between different curated IDP databases. So the starting point is always the same, meaning that these are databases which curate the literature one way or the other, and they extract information about the intrinsically disordered regions. And as you can see, the sizes of the databases vary, but the key point here is that at the center of all of them, there is virtually no information. So everybody's taking care of different examples. So there is a lot of room for improvement in this, to pull in the data, and that would serve as a much bigger data set than each individual database. Moreover, each of these databases has a slightly different focus, and therefore it also means that you would enrich the full annotation because you can cover the different phenomena. And remember that IDPs are by definition not a single phenomenon, but it's rather a mixed bag of phenomena where you can annotate each of them separately and with slightly different nuances. So this is the lack of overlap from the curated IDP databases. And on the next slide, what we can see here is actually some work we have been doing recently, where you see the overlap between DISPROT, which is the largest curated IDP database we're hosting, and IDEAL, which is another one in Japan, which is quite large. And surprisingly, the amount of information about long disordered regions with missing residues you can get from the PDB. And it turns out that the PDB is actually a major source for, through their long stretches of missing residues, is a major source for disordered regions. We have been validating a subset of these, curating about 100 of them, and in over 85% of cases, these are true disordered regions where the authors actually say it in the paper that this is a long stretch of missing residues precisely because it is intrinsically disordered. So it also shows that over the years there has been a change where even the crystallographers now feel more comfortable about saying that something is intrinsically disordered. And so there's a wealth of information which can be extracted from the PDB itself. As you can see on the right side, there's a distribution of the length of these regions. Now, the curated database are enriched for the very long disordered regions. and the ones we have from the PDB tend to be on the shorter side, so between 20 and 100 residues, more or less. But even that relatively small fraction of very long regions is still a significant number. And so this is quite a wealth of data. So I hope, on the next slide, please. I hope I have convinced you that there is a lot of data out there and that this should be pulled together in a way. So IDP annotations are so far significantly underrepresented in the ELIXIR core data resources. And integrating the IDP data resources can provide a blueprint for the integration of further IDP data in the core data resources themselves. You've already heard from Samir's talk earlier that through an implementation study we already had, we have been experimenting with integrating MobiDB Lite, which is just the annotation. It's a consensus prediction for intrinsic disorder. This has been pulled into Interpro, so it's available as data there. And from there, it has been propagated into UniProt and the PDB Europe. So this is a way that the information can be percolated into the core data resources. And in order to achieve this, what we need is on our end to be able to unify all the data into a single resource, which can then interact with the ELIXIR core data resources. Next slide, please. So the first step towards this is the MIADI standard, so the minimal information about this disorder experiment. And the starting point here is that there are no guidelines or standardized formats at all for sharing structural IDP data. So the MIADI guidelines are there to define really the minimum level of information that is required to comprehend the results of an experiment investigating the structure of an IDR. And in order to achieve this and to make this into a true standard, one of the things we have been doing, and this is especially the work of Balint Mesarosh and Norman Davey, is to work with the HUPOPSI, so the Human Proteome Organization Proteomic Standards Initiative, to create an intrinsic disorder work group where the details of the standard are being developed. And this was again possible thanks to the ELIXIR implementation study for the integration and standardization of IDP data. So next slide, please. The first step here, which is also the most fundamental, which has been already carried out, is to define the use cases. So set of representative disordered experiments we need to store, define the minimum information about the disordered experiments themselves, define the PSI-ID exchange format, both XML and TAP format to hold the MIADI standard, and define the controlled vocabulary required for the exchange format, and then to implement it in the core IDP resources, which is the point where we are right now. So the details and the report regarding the MIADI standard in its first incarnation can be found at the links shown there. And you will hear more about MIADI during the next talk by Zsuzsa, who will be talking about the ontologies, which are then the following step. So next slide, please. So the idea for the IDPcentral consortium is to build a single interface which can then interact with the ELIXIR core data resources. So the MobiDB CDR collaboration provides the blueprint for this. And the HUPOPSI ID work provides the means to transfer data seamlessly between the resources. So this will be at the central point where the data will be moved then, or can be moved into the ELIXIR core data resource. On our end, what we need to ensure is that all the databases which are part of the IDPcentral consortium adhere to the MIADI standard and implement it in a way that we can easily exchange the data. So next slide, please. In order to achieve this, what we have agreed is that we will mimic the IMEX consortium, which is for protein-protein interaction data. So we will maintain a shared curation effort and a shared export format, coming up with a single data set and a single detailed curation model. So what this means is that the various databases will curate data which will immediately be available in IDPcentral, and the databases themselves will maintain copies of that data, which can then be enriched with additional information they are interested in, if they want to have it. But all the data will be stored in IDPcentral, And above all, this will also ensure the sustainability of this approach, because if, for instance, after some years, one of these databases will no longer be available, then the data will remain available through IDPcentral itself. So in order to do that, we are implementing a Psychic-like interface in order to combine the data from the different resources. This is currently work in progress. We have the consortium in place. It will span across the globe, actually, because we have both partners in the US, which would be the BMRB, the Biomass Bank, which is the main repository for NMR data, and we have the IDEAL database in Nagoya University in Japan. So this is where we currently stand. There is already a preliminary website, but we are going to implement the core functionality of this over the coming months. And with this on the next slide, I would like to thank you for your attention.